Welcome to this AnaBios webinar on advancing analgesic drug discovery with a novel translational strategy. I'm Andrea Ghetti, the CEO of AnaBios Corporation. This presentation will be about 25 minutes long, and we will have a Q&A session at the end during which you will have the opportunity to ask questions through the chat box. Traditionally, non-clinical drug discovery has been conducted using animal models under the assumption that the results obtained in these models are translatable to clinical findings. Unfortunately, this is very often not the case, as proven by the high failure rate of developmental studies in clinical settings. Specifically, in the case of pain drug discovery, the situation is further compounded by the patient population heterogeneity. In this study, for example, different groups of pain patients affected by polyneuropathy, radiculopathy, peripheral nerve injury, or post-herpetic neuralgia were profiled using quantitative sensory testing. And as you can see, there are clear differences across the different patient populations, but also within each group of patients, within each specific etiology, there are differences in sensory loss, thermal hyperalgesia, and mechanical hyperalgesia. So the question is, how do the preclinical animal models of pain map onto these very diverse patient populations? And unfortunately, we don't really have a good answer, which means that in most cases, the results obtained in animal models cannot help us predict which specific therapeutic indication could be most appropriate for a given drug. AnaBios has decided to tackle these translational challenges by introducing a new paradigm, which enables the study of drug potential efficacy and risk directly into human cells and human tissues at the preclinical stage. Now, there are some fundamental challenges in trying to utilize human cells and tissues for investigational studies. The first one is ensuring sufficient viability and quality of the starting material. The second is access that is consistent and high enough to ensure proper support of drug discovery program. And the third one is reproducibility.